Hello friends, welcome to our YouTube channel, and today we are here with another topic, and that is called as ablation experiment in the C. elegans. In today's lecture, we will restrict ourselves to the vulva formations and the anchor cells formations and its ablation experiments. Okay, so it has come to the notice that lots of question has already been asked the CSI examination. So we are here with some of the experiments which clearly shows the fate of the event after the evolution of the specific cell in the development of the vulva and the anchor cell. Okay, so we will take an account of some of the experiments which could be asked in the further examination. Till now, we have understood and we have studied about the axis formation and the formation of vulva cells, vulva precursor cells, and the formation of the anchor cells. Okay, to continue the lecture, let us understand here. We had the 6 vulva precursor cells, and out of those 6, the P6P was getting the more lin-3 protein, and due to those cell which is getting the more lin-3 protein was able to produce the primary vulva precursors. Okay, so what will happen? What will happen if the P6P cell is ablated? So let us continue our lecture here with ablation of the specific cells. Okay, till now in the wild condition we know that so there is an anchor cell. Suppose this is an anchor cell, and here P3P, P4P, P5P, P6P, P7P, and P8P. P3P, P4P, P6P, P5P, P5P, P6P, P7P, and P8P. After it get the lin-3 protein, after, suppose this is anchor cell, okay, this is anchor cell, and the P6P is getting the maximum lin-3 protein. These are the lin-3 protein, and gradually the concentration of lin-3 protein decreasing after towards the peripheral cells. So for this, these 3 are the peripheral cells, and these 2 cells are the peripheral. Means P3 to P5 is peripheral to the P6P, and here P7P and P8P is peripheral to the P6P. What we get here means the P6P is getting the maximum amount of the lin-3 protein that we have studied already in the last lecture. So this was getting the primary vulva precursor cells, and these were the secondary vulva precursor cells, and this was the tertiary vulva precursor cells, and this will lead to the formation of the hypodermis. Okay, same here, this was getting the secondary vulva precursor cells, and these were the tertiary vulva precursor cells, and this was getting the fate of the hypodermis. These things we have already studied in the previous lecture. Okay, in the previous lecture. So we will try here to ablate one cell. We will try here in this lecture. We will try to explain what could be the fate of the cells after the ablation of the particular cells, or what could be the fate of the structure after the ablation of the particular cells. Okay, suppose let us start with the P6P. First experiment is, suppose what will happen if the P6P cell is ablated. Okay, what will happen if P6P cell is ablated by laser. Okay, So if this cell, if P6P cell will be ablated, okay, with the help of the laser, means this cell P6P cell is ablated here. Suppose this is ablated, okay, then if this cell will be ablated, then what will happen? The amount of lin-3 protein is maximally getting or in interaction or in contact with the P5P or P7P. So in this case what will happen? In this case what will happen? The P5P or P7P will acquire the fate of the primary bulbar precursor cells. Any of these cells, either P5P cell and P7P cell, will acquire the primary bulbar precursor cells. So the structure could be like this one. Suppose this is the P3P. Suppose there is an arrangement like this one: P3P, P4P, P5P, P6P is ablated, P7P and P8P, and P6P is ablated. Okay, so what will happen here? Either this one will acquire the primary bulbar precursor cell, or this one either this one or this one will acquire the primary bulbar precursor cells. And if it acquired, if P5P is acquired, suppose if P5P will acquire the primary bulbar precursor cells, then this one P4P will get the secondary bulbar precursor cells, and P3P will lead to the formation of the tertiary bulbar precursor cells. Okay, means this will acquire the hypodermis. Okay, if P5P is getting, if P5P is getting the fate of the primary bulbar precursor cells, then the P7P will be secondary and the P8P will be tertiary, will lead to the formation of the hypodermis. Okay. Okay, if P7P or if P7P is acquiring the bulbar precursor cells, then this one will acquire, this P8P will acquire the secondary fate, and secondary fate, this one will be the first degree fate, VPC, and this one will acquire the secondary one, okay, and the rest one will acquire the formation of hypodermis. So we come to know that if P6P is ablated, then what will happen? Either P5P or P7P will acquire the bulbar precursor cells. Okay, if P5P will acquire the bulbar precursor cells, then P5P will be the primary bulbar precursor cells, and the cell which is adjacent to the P5P will acquire the secondary fate, and the remaining will acquire the tertiary fate, and that will form the hypodermis. Okay, means here in this case the P7P will acquire the secondary fate, and P8P will form the hypodermis, will acquire the tertiary fate of the bulbar precursor cells. Suppose if P7P is ablated, then what will happen? P8P will acquire the secondary fate, P5P will acquire the secondary fate, and rest other will acquire the formation of the hypodermis. Okay, so in another experiment, what we come to know about, if suppose if P5P is, what will happen if P5P is ablated? If P5P is ablated, okay, so if P5P is ablated, then if P5P is ablated, suppose if P5P is ablated, then what will happen? Okay, the P6P P will be the primary fate, okay, and the P5P is ablated, then what will happen? The P4P will acquire the secondary fate and the P3P will acquire the tertiary fate, that is the hypodermis. And the remaining, the P7P again will acquire the secondary fate and the P8P will require the tertiary fate, that is the hypodermis formation. Means the P5P will be replaced by the P4P. Okay. Now what will happen if P7P is ablated? What will happen if P7P is ablated? What will happen if P7P is ablated? Now let us understand here. If P7P, suppose if this one is ablated, then again this will be replaced. The P7P, the fate of the P7P will be replaced by the P8P. It will be replaced by the P8P. Means the P6P, the P6P will having the primary and the P5P will having the secondary, and the remaining will form P4P and P3P will form the hypodermis. Okay, and P8P, because the P7P is already ablated and the P8P will require the secondary fate, means it will form the secondary VPC. Okay, now let us come to the another topic, another experiment. Suppose if all is ablated, if all the cells are ablated, means what will happen? Till now we have gone through the one by one cell ablation experiment. Now what will happen if all except the P3P is ablated? Suppose this one is also ablated, this one also, this one also, this one also, this one also will ablated. Then what will happen? If everything is ablated, if every cell is ablated, then only P3P will acquire the fate of primary VPC. Means only the 3P3 will be responsible for the formation of the primary fate of the vulvoapical cells. Okay, if we say that what will happen? If we say what will happen if all cell is ablated, suppose if P3P is also ablated, then what will happen? Okay, so there will be no vulva formation. In any case, the P2P or P1P or here P9P or P10P or P11P. So these are the cells which is lying towards the adjacent of the P3P and the P8P, and in no condition this P2P or P9P will lead to the formation of the vulva. There is no condition for the formation of the P2P and P9P to acquire the vulva. And with this experiment, it was found that there is only P3P to P8P are only responsible for the formation of the vulvoapical cells, and hence it was clear from this experiment when all the VPC were ablated, there was no formation of the vulva, means it was a vulva-less. The organism was having the vulva-less. Now what will happen if the gonad is ablated? If the gonad in L1 an anchor cell prior to the L3 is ablated, then what will happen? In another experiment, let us understand about what will happen if gonad at L1 stage and anchor cell prior to 3, means before 3, L3 stage is ablated. Okay, means if the gonad is absent, if the gonad is absent, it is ablated and the anchor cell is also ablated, then what will happen? If it is, if the anchors, because the anchors is alone as responsible for and is enough for the formation of the fate of the vulva, but if it is ablated before the L3 stage, then what will happen? All the structure, all the vulva precursor cell will acquire the 3 degree of the BPC, means all those structures will lead to the formation of the hypodermis. Okay, so the P6P, P3P, P4P, P5P, P6P, P7P and P8P all will acquire the fate of the 3 degree. And they will all form the hypodermis. They will form the hypodermis. They will form the fate of the hypodermis. Now, let us understand about another experiment. What will happen? Now, let us understand what will happen. What will happen if the place of the gonad is shifted somewhere else? What will happen if the place of gonad is shifted with the help of DIG1 mutation? Then what will happen? If gonad is displaced by DIG1 mutation, okay, in that case what will happen? The position of 1 degree, or you can say the primary vulva, could be changed based upon the position of the gonad. Okay, means the place of primary VPC could be changed. Okay, so the primary the fate of the primary vulva precursor cells could change. Okay, now let us understand about another experiment, because Lin-39, there is a protein, that is the Lin-39. Every cell which is from P3P to every cell which is P3P, P5P, P6P, P7P and P8P is responding to the they are responding to Lin-39 protein. Okay, and this is responsible for the first. What will happen? What is the function of the Lin-39? Actually, the Lin-39 stops the vulva precursor cells to fuse with the hypodermis first, and it also involves in the cell development. Means after the cells will acquire their specific fate, after this cell will acquire their specific fate, they will keep on dividing. Suppose this P6P, they will keep on dividing, and they will keep on dividing, means they will acquire this P6P will acquire the, after division they will acquire this P6P anterior and this P6P posterior. And what is the function of actually the Lin-39 is actually what does the Lin-39 does, that it will stop these two proteins, these two cells to acquire the structure of the hypodermis or to fuse with the hypodermis first. Second, it keep the pace of the division. Lin-39 keep the pace of the cell division. Okay, so what will happen if this Lin-39 mutation in the Lin-39 protein is there? Because it is secreted by, it is expressed by all those cells which is from P3P to P8P. Then what will happen if there is a mutation? Okay, so if there is a mutation, then what will happen? If there is mutation, if there is mutation in the lin 39, okay, if there is a mutation in the lin 39, then what will happen? All the VPC will fuse to form hypodermis, because each and every cells were being stopped to form the fate of the hypodermis, and it was led with the help of the lin 39 first, and the cell division was also promoted with the help of the lin 39. But if there is a mutation, if there is a lack of or there is suppressed secretion of the lin 39 is there, then what will happen? So all those VPC will acquire the fate of the 3 degree, and they all will form the VPC. They will all form the hypodermis. Means every cell, each and every cell of the vulva precursor cells, that is 3p3, 3p3, 3 p3, p3p, p4p, p6p, p7p, p8p, all will acquire the fate of 3 degree, and they will fuse with the hypodermis. Suppose what will happen if the anchor cell is ablated? If the anchor cell is ablated alone, okay, then again what will happen? There will be a formation of the 3 degree, means all the cell, all the VPC, all the vulva precursor cell will acquire the formation of the hypodermis, means they will acquire the formation of the tertiary vulva precursor cells, and they will form the hypodermis, or they will fuse with the hypodermis. So this was about some of the experiment regarding the cell ablation in context to the anchor cell and the vulva precursor cells. Hope you like the video. If you like it, please don't forget to share, like and subscribe. Thank you. Thanks for watching.